Hello guys, so let's continue with our glycolysis lecture. We are discussing glycolysis, and we have covered first step, which is one of the very important step. Okay. Now, yes. Now this glucose 6-phosphate is kind of chained. Correct? There is one very simple step now, because next step is very important, and this step is kind of reversible step. Okay, this step is reversible step. What is happening here? Here there is just isomerization which is happening. What is isomer of glucose? The glucose ka isomer is kind of a fructose. So from glucose 6-phosphate we still have 6 carbon sugar over here, 1 2 3 4 5 6. Okay, there is still a phosphate group. Okay, but just structure of glucose is little bit changed. Now it has attained a fructose structure. Okay, so this will be fructose 6-phosphate. Okay, if you want to see the structure, you will very easily able to see this structure. See, when this is, let's say 6, 1 2 3 4 5 6 carbon. This is our glucose ka structure which I am drawing here. Okay, this is glucose and this is OH. Okay, and here you have OH, OH. Here you have OH. Then you have H2, and we have attached here also a phosphate group. So here will be phosphate group. Okay, here there will be a phosphate group. And of course, we should not forget this side also, which is H, 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 OH, H. So this is our glucose molecule. Okay, 6 carbon sugar. And if you pay a little bit attention over here, you have kind of aldehyde group. Okay, here you have aldehyde group. Now what is happening in isomerization is, there is one simple thing which is happening over here. Okay, what is simple thing? This aldehyde group will be converted into a ketonic group. So 1 2 3 4 5 6, और बाकी सब ज़्यादा कुछ चेंज नहीं होगा. ओके, ज़्यादा कुछ बाकी कुछ चेंज नहीं होगा. So this is what is happening. Now it was aldehyde group and this is your ketone group. Correct? Just थोड़ा सा isomerization हो गया, glucose to glucose. This is glucose 6-phosphate, and this is fructose 6-phosphate, and this reaction is irreversible. Okay, if huge amount of glucose 6-phosphate is there, it will convert into fructose 6-phosphate. If fructose 6-phosphate is more, then it will convert into glucose 6-phosphate. Okay, and the enzyme, it is doing isomerization, so enzyme has to be isomerase. Okay, and what is the isomerizing? Glucose 6. So it is glucose, or you can also call it, this molecule is phosphoglucose, so you can call it phosphoglucose isomerase. It is phosphoglucoisomerase. Okay, so as I told you, this is not a very crucial step, glucose to the glucose 6-phosphate and fructose, glucose 6-phosphate to fructose 6-phosphate. Okay, because it is reversible. Now the next step, which is fructose 6-phosphate to, now what we are doing, we want fructose 6-phosphate to get converted into. So here what is happening here, very interesting thing which is happening over here. Okay, here your fructose 6-phosphate, okay, here your fructose 6-phosphate is converting into, let's say this is your fructose. 1 2 3 4 5 6. Okay, here there was one, this, what is this? Phosphate group. Okay, this fructose 6 phosphate is converted, and there is an enzyme over here which is very important enzyme. Okay, this is very important enzyme, one of the very important enzymes, or one of the very important steps. Okay, and the enzyme over here is, okay, very important enzyme, which is this was, so here it was phosphoglucoisomerase. Okay, phosphoglucoisomerase. Let me write it nicely over here. Phosphoglucoisomerase. Okay, and this enzyme is, so this is phosphoglucoisomerase, this is phosphofructose. Okay, and this enzyme also attaches a phosphate group at this molecule, so this will be kinase. So this molecule is phosphofructokinase. This is phosphofructokinase, and this step is irreversible. This step is irreversible. Now, if this step is irreversible, yeah, we have to attach one more phosphate over here. This is one more phosphate group. So here we have phosphofructokinase, and it is fructose 6 phosphate to, this is now here. What is the name of this molecule? The molecule is fructose phosphate. Kidhar hai? This is first, second, third, fourth, fifth, sixth. So at which position you have phosphate? It is here you have fructose. The molecule is called fructose 1,6 bis phosphate. It is fructose 1,6 bis phosphate. And this fructose 1,6 bis phosphate is very important also. Okay, and this enzyme is phosphofructokinase type 1. Now you will ask that why type 1, means there is a very high possibility that there is something called type 2. Yes, we will discuss that, what is type 2. Now when we talk about the regulation of this enzyme, of course here what it is happening here what is happening here ATP is converted into ADP. See, phosphate यहाँ पे आया ना, यहाँ पे phosphate नहीं था, so इधर phosphate आया. Phosphate कहाँ से लिया? We have taken it from ATP. So we'll ask, sir, हम लोग we want energy, but we are investing energy over here. Yes, we have to invest some energy to get some energy. So like that, this is the first step. But no, this is not the first step. Sorry. Here also, see, here also there is one phosphate. So of course here also there will be ATP to ADP. ATP to ADP. So here also hexokinase and glucokinase takes phosphate from ATP, it converts into ADP. Here also fructose, phosphofructokinase type 1 takes ATP and converts into ADP. So this phosphofructokinase has two kinds of arms. From one arm it takes your fructose 6 phosphate, it takes ATP, it converts fructose to fructose 1,6 bisphosphate and ATP to ADP. Okay, exactly like that, hexokinase and glucokinase, they take glucose, convert into glucose to glucose 6 phosphate, and they take ATP, convert ATP to ADP, and they transfer phosphate group for one molecule to another molecule. So here we have invested already 2 ATPs already हम लोग का चला गया है. Okay, so already we have invested 2 ATPs over here. Okay, and this step, let me tell you, this step is very important because this step is irreversible, this step is highly regulated, third, and this step is the committed step of glycolysis. See, when someone is committed, correct? What do you mean by commission? What do you mean by someone is committed? That means that person will do that thing. For example, a guy is committed to a girl, or a girl is committed to a guy. What is the end result? They had to get together in the future. So there is no other bifurcations. So if this step happens, if from fructose 6-phosphate to fructose 1,6-bisphosphate happens, now this pathway will go only and only in glycolysis. There is no other bifurcations. It will only go in glycolysis. For example, see, there are chances that your glucose 6-phosphate can go into HMP pathway. We will discuss what is HMP pathway. This glucose 6-phosphate can also go in glycogen synthesis. Glucose 6-phosphate is actually converted to glucose 1-phosphate, and then it is converted to UDP glucose, and then it goes to glycogen synthesis. Okay, so glucose, and also it can go to into glycolysis, correct? But if this step happens, there is no turning back. Then if this step happens, it has to go and make pyruvate. Okay, that's why this step is very important. This is committed step. We call this steps as a committed step. Okay, so let's understand what is the regulation of this enzyme. Okay, now this enzyme is regulated, of course, by, see, it has some pockets over here, correct? It has some different pockets over here. If you talk about activation, or this guy comes over here and activate, which is AMP. So AMP is activator for this enzyme, correct? Then even your, what is this? Even your fructose 6-phosphate is also an activator. For this enzyme. Okay, fructose 6, because see, अगर fructose 6-phosphate ज़्यादा बन रहा है, that means there is a signal we need to do glycolysis over here. So let's stimulate this enzyme. Okay, let's do more kinase activity to this fructose 6-phosphate. So fructose 6-phosphate is kind of an activator over here. Okay, then there is one more extremely powerful activator, and this extremely powerful activator, okay, this extremely powerful activator is 4, 5, 6. Okay, what is this? This is fructose 2,6-bisphosphate. This is fructose 2,6-bisphosphate. Now you will ask, sir, ये कहाँ से है? From where fructose 1,2-bisphosphate is coming? We know how fructose 1,6-bisphosphate is coming. It's from phosphofructokinase type 1. But from where fructose 2,6-bisphosphate is coming? This is also coming from this pathway only. So what our cell does, what our enzyme does, if there is lots and lots of fructose 6-phosphate, okay, it will convert it into fructose 1,6-bisphosphate, no doubt, but some amount it will also convert into fructose 2,6-bisphosphate. But we need enzyme for that, and the enzyme here I am drawing little bit. We are taking this fructose bisphosphate and converting into fructose 2,6-bisphosphate. समझ में आ रहा है? And the enzyme for this, okay, and the enzyme which we needed for this is very interesting enzyme, and this enzyme has two phases. Okay, it has two phases. One phase is and this is another face. Let's say this is a girl, and this is a guy. Okay. So this enzyme, this is a couple enzyme. Okay, this is a couple enzyme, and the couple enzyme is very interesting. This is actually two different enzymes. Okay, this two different enzyme is in one collaboration. Okay, for example, if we talk about this, like this, so we have two different activity which can be done by this enzyme. Okay, so what is this activity which we are talking about? So this, let's say this girl has this girl has kinase activity. This girl has kinase activity, and this guy has phosphatase activity. Phosphatase activity means one is giving kinase, other is removing, one which is kinase which is giving phosphate, this is removing phosphate. Are you understanding? They both differ in their action, but they are like every couple, they both have different actions and different moods, but they work together. Okay, so of course when kinase is happy, our phosphatase will not be happy. Okay, and vice versa. Okay, it happens normally everywhere. In a couple, at least one is happy and it makes another unhappy. Yes, so this is the situation over here. So when this is activated, kinase is activated. Okay, when this is activated, this will convert fructose 6-phosphate into fructose 1,6-bisphosphate. So when this is active, when this domain is active, the kinase domain is active, it will convert fructose 6-phosphate into fructose 2,6-bisphosphate. If phosphatase is active, it will remove the phosphate group, and it will again convert into fructose 6-phosphate. Let me tell you, it is not a reversible enzyme, guys. These are two different enzymes. Okay, these are two different enzymes working together. Okay, so it's not a reversible thing. One is, of course, दोनों एक साथ नहीं हो सकते. Either your kinase is activated when it will move fructose 6 to fructose 2,6-bisphosphate, or phosphate is activated which will again make fructose 2,6 to fructose 6-bisphosphate. And what is the advantage of making fructose 2,6-bisphosphate? Because this is master regulator of fructose phosphofructokinase 1. Okay, this is master regulator. That it will increase the activity of this enzyme by many folds. ये पहले enzyme कुछ 2 molecule ले रहा था and taking 10 molecules of fructose 6-phosphate and converting 10 molecules of fructose 1,6-bisphosphate. But when fructose 2,6-bisphosphate comes, now it can take 100 molecules of fructose and it can convert 100 molecules of fructose 1,6-phosphate. So the enzyme activity is increased over here. Okay, and who is this? This is this enzyme, our couple enzyme. What is the name of this couple enzyme? Yes, what is the name of this? We have not decided the name yet. And the name of this enzyme is phosphofructokinase type 2. I remember I told you this is type 1 and there will be type 2, and this is the type 2. So this is phosphofructokinase type 2 enzyme. Phosphofructokinase type 2 enzyme has two groups. One, it has kinase enzyme working with it, and one phosphatase enzyme working with it. Okay. So let us see when what will be activated. Okay. Yes. So when we talk about, you remember we have talked about glucagon. Yeah, glucagon is here. Glucagon has Glucagon has 7-pass receptor. Glucagon has 7-pass receptor. It has GS. GS will activate G alpha. G alpha will activate adenyl cyclase. I hope you know this pathway. Adenyl cyclase will convert ADP to CAMP. CAMP will activate pKa. Now this is very interesting. When pKa is activated, okay, when pKa is activated, pKa adds phosphate group to kinase. And when you add phosphate group to kinase, kinase is not happy. Kinase doesn't like this change, this charge. Kinase is very silent enzyme. It likes to work without phosphate. pK will add phosphate group to this kinase, and when it adds phosphate group to this kinase, kinase group becomes very unhappy. It doesn't want to give kinase to fructose glycinate, and of course when she is unhappy, he becomes happy and he is activated. And when he is activated, he will start removing the phosphate group. When he is starting removing the phosphate group, your fructose 2,6-bisphosphate will be converted into fructose 6-phosphate, and it will not complete the committed step because the master regulator is not there. Understand it? So when, as we have discussed earlier also, that glucagon receptors are main receptor which you found in liver. Okay, do you want liver to work to do you want liver to run the glycolysis cycle when there is less blood glucose? वो already rich है. Do you want it to spend more money when outside there is hunger? You want liver to give some food to everyone, which is the job of this. Okay, which is the job of our liver. Correct? Exactly like that. Exactly like that. When there is glucose, you don't want, when there is less glucose, and when who says that there is less glucose in the blood? Glucagon says. When there is glucagon, when it comes into the liver, it gives the liver a signal, see, blood glucose level is very less, you need to stop going into glycolysis. Okay, and you need to use some other source to make glucose and run your pathway, but give this glucose to another so that other tissues may work fine and the blood glucose level will be maintained. So in this pathway, what is happening? When glucagon is there, the kinase, it will add phosphate group to kinase, domain of phosphofructokinase 2, and this will become inactive. The whole enzyme, this will become inactive and this becomes active. Okay, and no conversion of fructose 6-phosphate to fructose 2,6-bisphosphate will be happening, and then it will not stimulate our phosphofructokinase 1, which will not convert fructose 6-phosphate to fructose 1,6-bisphosphate. Similarly, there is another enzyme which tells, okay, there is high blood glucose. Okay, there is high blood glucose in there is happiness outside in the blood. Lots and lots of amount of glucose. Liver, please don't send more glucose over here. Use how much glucose you want. And which is that enzyme who is bringing some happy news? And that hormone is insulin. Insulin comes when insulin binds to the receptor. Insulin has 1-pass receptor. Okay, through some pathway. Okay, it will add a phosphatase. And this phosphatase will remove this phosphate group from here. And when this phosphate group is removed, kinase will become again happy. And when kinase becomes happy, this becomes unhappy. And when kinase is happy, what kinase does? Kinase will convert. Now kinase is happy. Kinase will make fructose 6-phosphate to fructose 2,6-bisphosphate, and with some amount of fructose 2,6-bisphosphate can be a master activator of fructose phosphofructokinase type 1, and it can make lots and lots amount of fructose 6-phosphate to fructose 1,6-bisphosphate. So are we understanding what we are saying? Not only this, see, this molecule which is which is fructose 2,6 bisphosphate. This can regulate this enzyme also positively. We need more fructose 2,6 bisphosphate so that we can activate more phosphofructokinase type 1, so that we can make more fructose 1,6 bisphosphate, so that we can move our glucose into glycolysis cycle. Are we understanding what is this? Okay, is it very clear? Correct. So what is this so what are the activators when we are talking about? There is one more activator of this enzyme. I forgot. There is one more activator of this enzyme, and this activator is citrate. Citrate. What is citrate? Okay, I'll give you some very small introduction. When this glycolysis is running, glycolysis is converted into pyruvate. This pyruvate will go into Krebs cycle, and there is one product that is of Krebs cycle is citrate. Okay, there is one product which is citrate. Correct. When we have lots and lots of citrate, you need more Krebs cycle to work? You need more glycolysis to work? No, because citrate is saying, okay, we have lots of citrate now. Okay, then if you have lots of citrate, you do not need to do this glycolysis cycle now. So please stop the glycolysis. So citrate is kind of an inhibitor. Citrate is kind of a negative regulator. Correct. So who is activator over here? Activator is AMP. Okay. Master activator is who is your master activator over here? Your master activator is your activator is AMP. Your activator is fructose 6 phosphate. Okay. Your inhibitors are citrate. Okay. Then your inhibitor are ATP can also inhibit. ATP can also inhibit this. Okay. ATP can also, let me write here very easy, very nicely here. So you will understand. So what activator? Activators we have discussed AMP, fructose 6 phosphate, and who is master activator? Master activator is fructose 2,6 bisphosphate. Who is inhibitor? Inhibitor you have ATP and citrate. Correct. And who is extrinsic regulator? Of course, glucagon and insulin. Okay, glucagon can also come and activate this deactivate this. And who is activator and deactivator of this phosphofructokinase type 2 is, it's the similar what we have discussed over here. Okay, similar activator and deactivator over here also. And also see, in the starting also when I was talking about ATP to ADP ratio. So when ATP to ADP ratio is more, so of course the glycolysis should decrease. Okay, the glycolysis should decrease, and if ADP to ATP ratio is more, the glycolysis should increase. Exactly what that is happening over here. Okay, I hope you understood this step. This is very important step. So we will meet in the next video to discuss some more steps like this. Okay, yes. Thank you very much.